So, good afternoon students. So, today will be the last class of your biomolecules chapter. Again today also we are going to deal with biology la biomolecules, isn't it? Already 2 sessions has got over and today is the third class where we are going to complete this chapter. Okay. So, biomolecules, and today the left out topic is about the enzymes. We will see what are all the topics, subtopics that we are going to cover today. With that, the biomolecules chapter will be getting over. You will see that subtopics of this chapter, that is so the chapter name is what? Biomolecules. Under this chapter today we are going to study about the metabolic basis of living, the living state, then these are all small topics: chemical reaction, the last one is about the enzymes, its types, basic types and its action. Then what is meant by activation, energy, what is meant by inhibitors, enzyme inhibitors, it is meant by enzyme inhibitors and factors affecting enzyme action. Okay. So, these are the topics that we are going to see today. It's a very simple topic, we can complete it faster. So, with this the lesson will be getting over. Understood? Now, we will start with metabolic basis of living. So, in my previous class I have told you what is meant by metabolism. What is meant by metabolism? So, what is metabolism? The normal chemical reaction, taking place inside the body, of living organisms are called as metabolism, isn't it? So, because of this metabolism only the normal growth and development takes place. The normal growth and development takes place. Normal growth and development occurs, or otherwise normal growth and development takes place. When you take this metabolism can be broadly classified into 2 categories. One we are calling it as anabolism, the other one is called as catabolism. Anabolism and catabolism. So, anabolism, building up. Anabolism means what? Building up. What is this catabolism? Degradation. What is this catabolism means? Catabolism means degradation. If for say for example for anabolism, simpler substances combines to form larger substances, bigger substances, larger macro substances, we are telling no, larger substances. Say for example, the acetic acid combines to form cholesterol. So, acetic acid is a simpler substance these simpler substances like acetic acid will be getting combined and it forms a larger substance. The larger substance is a cholesterol. Now you come to this catabolism. Catabolism, the larger substance or molecules breaks down to form simpler. To form simpler substances, then we call it as what? Catabolism. In the very best example, we can take glucose to form lactic acid. To form what? Lactic acid. Glucose to form lactic acid. Glucose is 6 carbon compound. Lactic acid is 3 carbon compound. So 6 carbon compound to 3 carbon compound. Then the bigger molecules. This is the smaller molecules which takes place in the respiration. Now physiology we will be studying. Respiration la padipom. Respiration la will be studying Krebs cycle, glycolysis pathway la we will be studying all these things. Okay. So glucose to form lactic acid. So this is about the metabolism. Why all this metabolism and catabolism has to take place? Because it is a kind of a chemical reaction. As a result of this catabolism and the anabolism, the chemical bond will be getting formed inside the body. I can say this chemical bond as energy also. Okay. So now this chemical bond gets converted, or otherwise it will be getting utilized. It will be getting converted, or otherwise it will be utilized as energy. It will be getting converted, or otherwise it will be getting utilized as energy when during biosynthetic pathway. During the biosynthetic pathway. What is that biosynthetic pathway? The biosynthetic pathway is either respiration, transpiration, plants la photosynthesis, circulatory system ನಮ್ಮ ಒಡಂಬಲ ಅದೆಲ್ಲ biosynthetic pathway, and for osmotic, and finally for osmotic process, osmotic regulations la, and for our mechanical work, and to carry out the various mechanical work which is needed. The energy is needed. How this energy is getting produced? From the chemical bond. How this chemical bond restored inside the body? Due to the chemical reaction. What is that chemical reaction is called? Chemical reaction is called as metabolism. What are the two types of metabolism? Anabolism. The other one is called as catabolism. Okay. So again we will revise. What is this metabolism? So for a normal individual person, for the living beings to sustain, to grow, the body mass index has to get matched with that of the height. So everything should be normal, appropriate. It should be in a proportionate manner, well defined manner. Enadella well defined manner arkano, the height, the body features and the weight, the mass of the body should also be appropriate. If it is not going to be appropriate, then what will happen? That results in disfiguration. Disfiguration, malformation should not take place. So the malformation to overcome, disfiguration to eradicate, the metabolism plays a very significant role. So this metabolism is nothing but it is a kind of a chemical reaction which takes place inside the body of all the living organism in order to sustain with the normal growth and the development. That chemical reaction is of two types. One is anabolism and catabolism. Anabolism means building, so combining together. So micromolecules combines together, giving rise to the formation of macromolecules. We are calling it as what? Anabolism. What is this catabolism? The heavier molecules or macromolecules will get splits up into two simpler molecules, then we are calling it as what? Catabolism. So we have anabolism and catabolism. The examples also I have given you. Why this type type of chemical reaction has to take place inside the body? The chemical reactions like this, metabolism, anabolism and catabolism, is very, very essential inside the body in order to produce various chemical bonds, and such type of chemical bonds will be restored in the various parts of the body. And whenever there is a mechanical work, osmotic regulation and biosensory pathway occurs, the chemical bond will get transferred in the form of an energy. That energy is called as what? We call it as ATP. ATP is called as what? Adenosine triphosphate, which is the energy currency of the cell. What is ATP? ATP is adenosine triphosphate, which we call it as the energy currency of the cell. So the energy has to be getting produced, energy currency of the cell. So how this energy will be getting produced? Because of this metabolic reactions, metabolism. Okay, that will be our first step. Now we go to the second topic. What is that second topic? Chemical state. What is metabolism? What are the types of metabolism, and why the metabolism should take place inside the living organism? Ultimate another to get the energy in order to carry out the various work. So metabolism helps in the liberation of energy. Either it may be an exothermic or otherwise it can be an endothermic. Usually it will be what? Exothermic process. Now we pass it on to the living state. We know there are varied living organisms existing in this world, isn't it? Different types of living. Biodiversity you would have studied. So the living state. The living state. So there are varied living organisms, varied plants and animal features existing in this world, existing in the world, isn't it? And each and every living organisms of plants and animals, which starts from unicellular to multicellular, prokaryotic to eukaryotic level, cellular grade of organization to tissue level grade of organization. What type of organisms you take, all the organisms will be made up of biomolecules. All the organisms, irrespective of the cellular formation or tissue formation, irrespective of the classification, irrespective of its growth and the development, all the living organisms from primitive to the advanced level of organisms, all the organisms are made up of what? The biomolecules. They are all made up of biomolecules. Made up of biomolecules. See, you can find out lots of variations in the morphological pattern of plants and animals. Morphological pattern there will be lots of contrasting features. Anatomical characteristic features you can find out the varied different aspects, contrasting characteristic features. Physiological aspects, when you take the physiological aspects of the living organism, also you can find out the contrasting character differences will be there. So physiological features, morphological features, anatomical features, which are the main criteria for the formation of the living organisms. Idella romba important. Anatomy is an internal feature. Internal structure should be formed appropriately. Morphological pattern, external features, external arrangements of various body features, morphological patterns we are telling. Physiological aspects, the functioning of the various organs inside the body. So physiology differences plants and animals. In morphological pattern also there is a varied differences. In anatomical features also there is a varied differences. Though there are varied differences depending upon the unicellular and the multicellular level of organizations, varied differences occurring in the physiological, morphological and anatomical features, but the biomolecule aspect of the living organism tends to be same. The biological aspect. So when there is a biological or biochemical aspects is going to be the same. Biochemical aspects are going to be same, made up of biochemical. So the biochemical or biomolecular features tends to be same for all the living organism, for all the living organisms. Biomolecular features tends to be same for all the living organisms. So when the biomolecular feature is going to be same, then that results in a steady state. That results in the steady state. எதுல steady state? அந்த chemical, biochemical arrangements inside the body. Arrangements inside the body, there will be a steady state. When there is a steady state, automatically நான் ஏற்கனவே சொன்ன மாதிரி metabolism occurs. When there is a steady state, it results in normal, previous topicல சொன்ன மாதிரி normal metabolism occurs, isn't it? The normal metabolism takes place. When there is a normal metabolism, then it will result in the normal growth of the individual. Normal growth of an individual. Normal growth of an individual. Now you have to keep one thing in your mind, that is, whenever the organism is undergoing a steady state, whenever the biochemical or biomolecular feature of an organism tends to be in a steady state, then that results in the non-equilibrium condition. Non-equilibrium condition. So even for in chemistry also, the reactant molecules will react with one another. When the reactant molecules will react with one another, when the organism did not attain the stability, the equilibrium state. So in order to attain the stable equilibrium condition, the reactant molecules will react with one another to get a product. When it attains the product, then automatically it will restore its equilibrium and stability. இப்போ நம்ம கூட when we are feeling hungry, then we will eat in order to compensate our hunger, isn't it? When we are not feeling hungry, when we are not feeling thirsty, obviously we will be under a state of equilibrium. We will not feel like drinking water or eating, isn't it? Similarly for a chemical reactants also, when the reactants are fail to attain the stability, unstable nature, if the reactant molecules are going to exist, then those reactant molecules have to react with one another to give rise to the formation of a stability, or in order to attain the equilibrium condition. So we also in day-to-day activities, we tends to be in a state of equilibrium, in a balanced state. Equilibrium condition நம்ம attain பண்ணணும், then only we will sleep. Our mind will be under a state of rest. If we have not attained the stability, or if we have not attained the state of equilibrium, dynamic equilibrium condition, then again we will start to work accordingly. எந்த ஒர்க்கை நம்ம கம்ப்ளீட் பண்ணலையோ, our mind will start to rattle about that particular aspect or the particular work, and unless until we are completing that work, we will not be in a state of stable. So we will be under a non-equilibrium condition. And similarly, when you take the living organisms, inside the living organisms, the biomolecules will be present, and these biomolecules will be under a steady state. So if the biomolecules, simply if it is going to be in a steady state, then what will be the use? Then that will not help the living organism to undergo the metabolism. Can you understand what I am telling? அப்போ ஸ்டடி ஸ்டேட்டில் வெறும் மாலிகுல்ஸ் மட்டும் பயோமாலிகுல்ஸ் மட்டும் ப்ரெசென்ட் ஆகிருக்குன்னு சொல்கிறோம். அப்போ எங்கே மெட்டபாலிசம் நடக்கணும்? த மெட்டபாலிசம் ஷுட் டேக்ஸ் பிளேஸ். அந்த ஸ்டடி ஸ்டேட்டில் இருக்கக்கூடிய பயோமாலிகுல்ஸ் ஹேஸ் டு அண்டர்கோ த கெமிக்கல் ரியாக்ஷன், ஹேஸ் டு அண்டர்கோ அ மெட்டபாலிசம், தென் ஒன்லி தேர் வில் பி அ நார்மல் க்ரோத். அப்போ அந்த மெட்டபாலிசம் போகணும் அப்படின்னு சொன்னாவே தட் ஷோஸ் த பயோமாலிகுல்ஸ் which is present inside the organism steady state la irukakudiyadhu is under a state of non equilibrium, under equilibrium or non equilibrium. When it will undergo the chemical reaction, when the molecules or when the biomolecules is not under a state of equilibrium, when it is under equilibrium or non equilibrium state, then the biomolecules will start to react with one another in order to attain the state of equilibrium. So appa non-equilibrium to equilibrium will ensure the metabolism to takes place rapidly. நான் சொன்னதெல்லாம் புரிஞ்சுச்சா? இப்போ என்ன நான் சிம்பிளாக திரும்ப again I am repeating for you, listen carefully. The concept புரிஞ்சிட்டா then you can write off your board. What I am telling, so there are varied categories of the living organism. Biodiversity la படிக்கிறோம். plants la எடுத்துக்கிட்டா எவ்வளோ வைரஸ்ல தொடங்கி வைரஸ், பாக்டீரியா, algae actinomycetes, lichens present ஆயிருக்கு. இப்போ கிளாஸிஃபை பண்ணி நம்ம கொண்டு வரோம். and then bryophytes, we have pteridophytes, gymnosperms la angiosperms la monocotyledonous varieties, we have dicotyledonous variety. Again in animals when you go, it starts from porifera la இருந்து coelenterata, அதுக்கு அடுத்தது annelida, arthropoda, nematodes இருக்கு. nematodes la platyhelminthes, ascalhelminthes இருக்கு, echinodermata, and then chordata la திரும்பவும் we have various classes இல்லையா, aves ல இருக்கு, amphibians இருக்கு, reptiles இருக்கு, pisces present ஆயிருக்கு. then finally we ourselves are the mammalian species exist ஆயிட்டு இருக்கும். இவ்வளோ classifications இருக்கு. இந்த classifications நம்ம எதை வச்சு கொண்டு வரோம்? based on physiology, morphological and anatomical and evolutionary characteristics based பண்ணி we are classifying the organisms. Though there are varied living organisms present, but the biomolecules present inside the living organism tends to be the same. எல்லா லிவிங் ஆர்கனிசத்தில் இருக்கக்கூடிய பயோமாலிகுல், கார்பன் எல்லா ஆர்கனிசத்திலும் இருக்கணும். கால்சியம் should be present in organic biomolecules. Organic biomolecules, hydrogen should be present. Though it is going to be a porifera mammalian species ஆங்கிறது it's not a factor. அது போரிஃபெராவை தொடங்கி மேமல்ஸ் வரைக்கும், வைரஸில் தொடங்கி ஏஞ்சோஸ்பர்மில் இருக்கக்கூடிய டைகாட்லிடனஸ் பிளான்ஸ், சிக்கோயே ஹையஸ்ட் டால் ட்ரீ அப்படின்னு நம்ம சொல்லக்கூடிய டைகாட்லிடனஸ் வெரைட்டியில் ஏஞ்சோஸ்பர்மிக் பிளான்ட். ஸோ ஹையஸ்ட் பிளான்ட் அப்படின்னு நம்ம சொல்கிறோம். அந்த ஹையஸ்ட் பிளான்ட்லேயும் பயோமாலிகுல் சேம் தான். வைரஸ் which we cannot see with our naked eye, only through the compound microscope இல்லை ஸ்கேனிங் மைக்ரோஸ்கோப்பில் தான் நம்மளால் பார்க்க முடியும்னா அந்த ஆர்கனிசத்துக்குள்ளேயும் அதே கார்பன் தான், அதே ஹைட்ரஜன் தான், அதே ஆக்சிஜன் தான். அப்போ there is no distinguishing or differences existing in the aspect of biomolecules. Only in the characteristic features there are various changes. ஆனால் biomolecules எந்த சேஞ்சஸுமே இல்லை. அப்போ எல்லா லிவிங் ஆர்கனிசத்தோடையும் the biomolecules are present. When the biomolecules are present, that shows the organism is under a steady state. வெறும் பயோமாலிகல்ஸ் மட்டும் if it is going to be present inside the living organism, will that be enough? No, that will not be enough. So the steady state எதை நம்ம டிஃபைன் பண்ணுறோம்? steady state when the biomolecules are present inside the living organism. When the biomolecules are present, then the living organism tends to be in a steady state. அப்படின்னு சொல்கிறோம். லிவிங் ஆர்கனிசம் டென்ஸ் டு பி இன் அ ஸ்டடி ஸ்டேட். ஸ்டடி ஸ்டேட்டோடு இருந்தால் லிவிங் ஆர்கனிசம் க்ரோ ஆகுமானா இட் வில் நாட் க்ரோ. ஃபார் தட் வாட் இட் இஸ் நீடட்? த ஸ்டடி ஸ்டேட்டில் இருக்கக்கூடிய பயோமாலிகுல்ஸ் ஹேஸ் டு அண்டர்கோ அ ப்ராசஸ் கால்ட் அஸ் மெட்டபாலிசம். has to undergo a condition called as metabolism. What is metabolism? The metabolism is a chemical reaction. So metabolism நடந்தால் தான் normal growth and development will takes place. இப்போ biomolecules equilibrium conditionல இருக்குமா or otherwise non equilibrium conditionல exist ஆகுமாங்கிறத question. What is that? Whether the living organism will be in the equilibrium condition or otherwise biomolecules எங்கே இருக்குது? Inside the living organism steady stateல இருக்குது. இப்போ steady stateல இருக்கக்கூடிய ஒரு biomolecules will be under a state of equilibrium or otherwise in a state of non-equilibrium. The question. The answer for this is the biomolecules will be under non-equilibrium. Non-equilibrium condition will be in the non-equilibrium condition. அப்போ தான் அதால் என்ன செய்ய முடியும்? Then it undergoes metabolism, chemical reaction inside the body, metabolism. Then it produces energy. அப்போ எனர்ஜி ப்ரொடியூஸ் பண்ண உடனே that biomolecules will be under a state of equilibrium. அவ்வளோதான் டாபிக். ஸோ நான் ஈக்குலிபிரியம் கண்டிஷனில் இருக்கக்கூடிய ஒரு பயோமாலிக்யூல் ஒன்லி வில் அண்டர்கோ தி மெட்டபாலிசம் கிவிங் ரைஸ் டு த ஃபார்மேஷன் ஆஃப் அன் எனர்ஜி அண்ட் அட்டைனிங் த ஸ்டேட் ஆஃப் ஈக்குலிபிரியம். Can you understand? So biomolecules ellame same kind of biomolecules, adula very much differences illa. For all the living organism will be having more or less same kind of organic and inorganic biomolecules inside the body. And the biomolecules mattum irundha poduma for the normal growth and development? No, that will not be sufficient. These biomolecules have to undergo rapid metabolism. Appo or biomolecule rapid metabolism kulla ponom appadina, it will be under a non equilibrium condition. Then only it will undergo the chemical reaction in order to attain the stable in order to attain the equilibrium state. Correct தானே? நான் ஈக்குலிபிரியமில் இருந்தால் தான் அதால் என்ன செய்ய முடியும்? It can attain the state of equilibrium. அப்போ ஒரு பயோமாலிக்யூல்ஸ் ஸ்டடி ஸ்டேட்டாக இருக்குது, நான் ஈக்குலிபிரியம் கண்டிஷனில் இருந்து it get transferred to the equilibrium condition after the metabolism. Okay, this will be the second topic. First topic நம்ம என்ன படிச்சோம்? Metabolic basis of living. Second topic what we have studied? The living state. Now the third topic is a chemical reaction. So chemical reaction pathi I don't want to give you lots of explanation for you. Instead we'll take one example. The water reactant, product, catalyst and all those things you will be knowing. So chemical reaction le for the chemical reaction, water reacts with carbon dioxide in the presence of an enzyme. Enzyme illa catalyst ni padichupom. Carbonic dehydrogenase. This is a catalyst. What is a catalyst? Catalyst is a substance that enhances the rate of chemical reaction, that speeds up the rate na speed na velocity of a chemical reaction, that enhances the rate of chemical reaction. So then it gives rise to the formation of a product, that is H2CO3, which is called as carbonic acid. Now so what is that is getting formed? Carbonic acid அப்படினு நம்ம சொல்றோம், carbonic acids ன்னு சொல்றோம். இப்போ you have to keep it in mind the role of a catalyst. நான் ஏற்கனவே சொன்னேன், catalyst will enhance the rate of chemical reaction. Suppose if this reaction is without a catalyst, what will happen? We'll see. Without a catalyst illa, then what will happen? The reaction can proceed and it can produce 200 molecules of H2CO3 in an hour. So ஒரு மணி நேரத்தில் it can produce 200 molecules of H2CO3 without the presence of a catalyst. Suppose in the presence of catalyst, with the catalyst or presence of catalyst, in the presence of catalyst, அப்போ நான் catalyst யூஸ் பண்ணுறேன், it produces 600,000 molecules of H2CO3 produces எவ்வளோ? 600,000 molecules of carbonic acid. H2CO3 is called as what? Carbonic acid per second. Up now you see, this shows what? Whenever the catalyst is used, the rate of chemical reaction increased to 10 times. Your simple reaction, water with carbon dioxide in the presence of carbonic dehydrogenase. So this carbonic dehydrogenase is a catalyst. It forms what? Carbonic acid. Now suppose with the catalyst only have written, without catalyst if the reaction is going to take place, even then it will be producing the carbonic acid, but the amount of carbonic acid produced will be 200 molecules of carbonic acid in an hour. So per hour it can produce 200 molecules, provided if I am going to use a catalyst, in the presence of a catalyst, Same carbonic acid will be getting produced, but how many molecules of carbonic acid? 600,000 molecules of carbonic acids will be getting produced per second. Can you understand how many molecules? 600,000. That means what? Whenever a catalyst is being used in a chemical reaction, the rate of the chemical reaction will be getting increased to 10 times. I can write one equation for this. The rate of a chemical reaction will be is equal to minus del concentration of R divided by, chemistry that is plus del into concentration of P divided by dt. You will be studying it in 12th standard. It is simplified. I can write as this reaction, I can write it in a simplified form as rate is equal to del, that is plus del into concentration of the product divided by dt, that is equal to minus del into concentration of R divided by dt. What is this meaning? This means delta, that is change in the concentration of the product divided by dt. Why I am writing plus here? That means there is an increase in the concentration of a product. So now you see, reactant reacts to give rise to the formation of a product. இப்போ reaction எதுமே நடக்கல. The reactants hasn't get react with one another to give a product. வெறும் நான் reactants மட்டும் எடுத்துருக்கேன். அப்போ what will be the concentration? 100%. What will be the concentration of the product? It will be 0% because reaction நடக்கல. Product எதுமே form ஆகல. அப்போ product ஓட concentration will be 0%. Concentration of the reactant will be 100%. அப்போ reaction நான் கொஞ்சம் கொஞ்சமா start பண்றேன். Start பண்ண உடனே என்ன ஆகும்? The concentration of the reactant goes on decrease, like it comes to 99%, 98%, 97%, 96%, like that. The concentration of the reactant will goes on decrease with the increase in the concentration of the product, like it will be in a 0%, then it comes to 1%, then it comes to 2%, then it comes to 3%. That means the concentration of the product will get increased with respect to what? With respect to the time period, that is t. Upper the concentration of the reactant goes on decreases. I am representing it in minus. The concentration of the product is getting increased. That I am writing it as plus. That is plus into change in concentration of the product with respect to time. The d is the change in concentration of the time will be equal to change in concentration of the reactant. Enna change? Decrease is the change. Minus is that implies what? There is a decrease in the concentration of the reactant with respect to time. அப்போ ஒரு கெமிக்கல் ரியாக்ஷனுக்கு கேட்டலிஸ்டோட ரோல் இஸ் ப்ரிடாமினன்ட். ஸோ த கேட்டலிஸ்ட் வில் ஆல்வேஸ் என்ஹான்ஸ் ஆர் த கேட்டலிஸ்ட் வில் ஆல்வேஸ் ஸ்பீட்ஸ் அப் த ரேட் ஆஃப் தி கெமிக்கல் ரியாக்ஷன் அப்படின்னு சொல்கிறோம். Okay, now the last topic which we have is an enzymes, about the enzymes. Now we will study in detail about the enzymes. ரொம்ப simple story will be the enzymes. We will study about the enzymes. So what are enzymes? Enzymes are the biocatalysts which catalyze various metabolic reactions taking place inside the living organisms. Are the enzymes. What are enzymes? Enzymes ingiradhu which is a biocatalyst which catalyzes the various metabolic reactions taking place inside the body. Which catalyze the various metabolic reaction taking place inside the body of the living organisms. So usually the enzymes are nothing but it is a protein. What type of protein? Majority of the enzymes are made up of globular protein. It is a globular protein. Okay, some enzymes are nucleic acid enzymes. Nucleic acid proteins or nucleic acid enzymes irukku. Such a type of enzymes made up of nucleic acids like RNA, adhaadu RNA enzymes அப்படினு சொல்லுவோம். They are called as ribozymes. Such type of enzymes are called as what? Ribozymes. Such type of enzymes are called as ribozyme. Now we go for a small classification to understanding purpose. I am writing. See, enzymes. When you take these enzymes, it will be having a protein part, and similarly it will be having a non-protein part. Non-protein part. Rendu part irukku. It will be having protein part and non-protein part. Ipo in the protein part, we can call it as apoenzyme, and this non-protein part we have, that we can call it as coenzyme. Apoenzyme, this we can call it as coenzyme அப்படினு சொல்றோம். Now this non-protein part, when you take, it is broadly classified into an organic part. The other one is called as an inorganic part. Now when you take this organic part, it will be having two categories again. One we are calling it as cofactor part. The other part is called as prosthetic part. Prosthetic part அப்படினு சொல்றோம். Inorganic, when you take inorganic constituents, நம்மளுக்கு ஏற்கனவே தெரிஞ்சது தான். என்னென்னலாம் இருக்கும்? It may have calcium, it may have iron, it may have cobalt, it may have zinc அப்படினு சொல்றோம். இத மாதிரி என்சைம்ஸ்க்கு பேரு conjugate enzymes அப்படினு சொல்றோம். Conjugate enzymes. See, when you take these enzymes are broadly classified into two types. One we call it as simple enzyme, the other one we call it as the conjugate enzymes. Simple enzymes, single type of enzymes will be present. The structure it will not be very complicated. Conjugate enzymes அப்படிங்கிறது two or more simple enzymes will combine together, giving rise to the formation of a conjugated enzyme. The simple enzymes can an example, you can write urease, you can write trypsin, or some of the examples of the simple enzymes. If in the conjugate enzyme we have apoenzyme part, we have coenzyme part, that is protein part. The other one we call it as non-protein part. The non-protein part we will be having an organic part, the other one is we call it as inorganic part. Organic part cofactor, the other one is called as a prosthetic factor. Prosthetic group, cofactor group அப்படினா where the enzyme molecules are very loosely arranged. இங்க prosthetic groupல பாக்கும்போது the enzyme molecules will be very tightly bound, then we are calling it as a prosthetic group. Inorganicல பாக்கும்போது calcium present are calcium ions, Fe2 plus ions, sometimes cobalt 3 plus ions, Zn2 plus ions, then nickel 3 plus ions will be present. இதெல்லாம் நம்ம என்னன்னு சொல்றோம்? We are calling it as inorganic constituents. இது just நம்ம தெரிஞ்சிக்க வேண்டிய enzyme பத்தி தெரிஞ்சிக்க வேண்டிய ஒரு சின்ன classification. Now we go for the action of enzymes. Enzyme action அப்படி நம்ம சொல்றோம். We go for the enzyme action. Enzyme action என்னன்னா, first we take the tertiary protein. நான் ஏற்கனவே புரோட்டீன்ல I taught you. Next topic is what? Other continuation is enzyme action. Enzyme action. So enzyme action first we will take the tertiary protein. What is the tertiary protein? Tertiary protein அப்படிங்கிறது it is a single polypeptide chain of secondary protein. This single polypeptide chain will be getting coiled around itself and giving rise to the formation of 3D structure is called as tertiary protein. Now previous classification protein primary structure, secondary structure, tertiary structure la sonne. So in a single polypeptide chain the single polypeptide chain is getting coiled around itself, giving rise to the formation of 3D structure. This is a tertiary protein. We have taken what? Tertiary protein. Now this tertiary protein will be having the tertiary protein will be having the crevices, or otherwise we can call it as a peaks. So tertiary protein has Another pair I can write it as crevices, edges, peaks, pockets. எந்த term வேணாலும் இது எல்லாமே synonyms. Any terms we can call. இத மாதிரி ஒரு peaks, crevices, edges will be present. And these crevices, edges, peaks and pockets are called as the active center. It is called as what? Active center, or otherwise called as an active site of an enzyme. Tertiary, நான் ஏற்கனவே example எழுதிருக்கேன். Tertiary proteins tends to be enzyme. Most of the enzymes are, how it will be? It will be in the form of a tertiary protein. அப்படின்னு சொல்கிறோம். It will be in the form of a tertiary protein. In the bookல, உங்களோட NCERT bookல, they have not given you in the detailed form. நான் in a simplified formல I am explaining you. Okay, as crevices, edges, peaks, pockets இருக்கு. That edges, crevices are called as the active site of an enzyme. So இது பேரு நம்ம active site னு சொல்றோம். First step. Next என்ன ஆகும்னா, இந்த active siteல the substrate molecules, the substrate, what is that substrate? The reactant molecules. Substrate na reactant molecules. The substrate molecules will get attached. அப்போ what you will get? Enzyme plus substrate complex உனக்கு form ஆயிடுச்சு. Enzyme plus substrate. So which giving rise to the formation of enzyme, substrate, complex. Now when the enzyme substrate complex is getting formed with, it is getting formed with chemical bonds, with the chemical bonds. Immediately the chemical bond breaks and the products from the active site of enzyme will be produced. அவ்வளோதான், இதுதான் enzyme action. என்ன இப்போ ஞாபகம் வைக்கணும்? enzyme actionல ஒரு protein enzyme எடுக்கிறோம். the enzyme will be having crevices இருக்கலாம், edges, pockets or peaks we are telling. That center is called as an active site. To that active site, the reactant molecules called as the substrates will comes and attaches, and it just modifies the shape of the primary, secondary, tertiary protein, and as a result the substrate molecules will get attaches itself towards the active sites by forming the chemical bond. Now once after the complex enzyme substrate complex is getting formed, then automatically the chemical bonds will starts to break, and from the active site the product molecules which will be getting formed will be getting eliminated out from the enzyme. So that is what we are calling it as enzyme action. We are calling it as what? Enzyme action. In the enzyme action alla tharava padichu vechukano, that you must be knowing. Okay, then we must know what is an activation energy nu oru concept irukku enzyme la. Activation energy is nothing but we call it as an additional energy. What is activation energy? Activation energy is nothing but we call it as an additional energy. Activation energy. Now see, we'll plot a graph first. Activation energy. Let me draw a graph. Based on that we will study. So this is reaction coordinate, or otherwise progress of the reaction. Progress of this is called as this axis, y axis, and we have x axis, isn't it? This is called as the potential energy. Now see, the reactant molecules are present here, and from the reactant molecules the reaction proceeds and it comes down and finally there is a formation of the product. Okay, now same, the reaction happens and it goes up and finally it comes down here. If I now listen the differences, what is the difference? You can notice ingirundu you can see the difference. Ingirundu inge you can see the difference. So here I am writing, this is an activation energy without enzyme. This region is the activation energy, the energy rich zone, activation energy with enzyme, and this is a transition state. This is also a transition state. Now you listen, this is the reactant and here we will be getting a product. But reactant molecules will react with one another. Can you see the reactant molecules react with one another? First peak, you see the first graph I am not giving the enzyme, without enzyme. Enzyme is a catalyst. I am not using the catalyst. So therefore, the reaction activation energy maximum is being utilized. Activation energy oda utilization eppadi irukku? Maximum poiduchu. Aprom it give rise to the formation of a product. Second step la what I have used? Activation energy with enzyme. So enzyme I have used. அப்படி enzyme நான் யூஸ் பண்ண உடனே எனர்ஜி யூட்டிலைசேஷன் கிராஜுவலாக என்ன இருக்குது, இங்கேயும் கம்பேர் பண்ணும்போது ஃப்ரம் ஹியர் டு ஹியர் இந்த அளவுக்கு தேர் இஸ் அ டிக்ரீஸ் இன் தி ஆக்டிவேஷன் எனர்ஜி, இஏ அப்படின்னு சொல்லுவோம். ஆக்டிவேஷன் எனர்ஜி இஸ் ரெப்ரசன்டட் பை த சிம்பிள் தட் இஸ் இஏ. ஆக்டிவேஷன் எனர்ஜி கோஸ் ஆன் டிக்ரீஸ். இப்போ எழுதுவோமா? என்ன ஃபஸ்ட் ஸ்டெப்பில் என்ன, ஃபஸ்ட்டு திஸ் இஸ் கிராஃபில் வரக்கூடிய ஸ்லோப் 1, this is slope 2, ரெண்டு இருக்கு. First we will write for the slope 1. Slope 1 la what is the condition? There is no enzyme. அப்ப என்ன இருக்கு? Maximum activation energy is used. Correct? Now when the enzyme is utilized, the activation energy utilization is less. Correct? Energy enzyme use panna activation energy is low. When enzyme is not used, when catalyst is not used, the activation energy will be maximum, which reaches to the transient state or transition phase, which attains the transition phase. Transition phase na further extension will not take place. This is the kadasi end point da nam enna solrom, transition. Furthermore the graph will not get exceeded. Furthermore activation energy utilization will not be permitted. Appadina da nam enna solrom, transition phase or transient state. Eppadi vena sollalam, transition state illa transition, transient, any terms we can use. Appo enzyme use panna namalukku activation energy will be very very low. Whenever activation energy tends to be low, the rate of reaction faster. Whenever the rate of reaction is faster, then that shows the reaction is spontaneous. Whenever the reaction tends to be spontaneous, such type of reactions tends to be exothermic reactions. There tends to be exothermic reaction. Again we'll revise. So na enzyme use panna utilization of activation energy na additional energy. What is activation energy? Activation energy means additional energy. edukku theva namalukku additional energy enga irundhu kudukuradhu? Additional energy for the reaction to takes place. adukku badal what we can give? We can use this enzymes or the catalyst. Appo whenever there is an enzyme is used, the activation energy utilization will be very, very low. Activation energy utilization கம்மி ஆயிருச்சுனாவே the rate of the reaction will be faster. The rate of the chemical reaction will be faster. Whenever the rate of the chemical reaction faster இருந்ததுன்னா then the reaction tends to be spontaneous. Whenever the reaction is spontaneous, that shows the reaction is exothermic reaction. அந்த reaction எப்படி இருக்கும்? It tends to be exothermic in nature. So அப்போ என்ன சொல்கிறோம்? Whenever the enzyme, a catalyst is used in a chemical reaction, the energy, activation energy utilization will be very low. Energy of utilization, activation energy utilization கம்மி ஆயிடுச்சுன்னா the rate of the chemical reaction will be faster. Faster the reaction tends to be spontaneous. Whenever the reaction is going to be spontaneous, then that shows the reaction is exothermic in nature. Okay. So this you have to study and you have to keep it in mind. The next topic is the factors, simple topics that I will tell you. The factors affecting the action of enzymes. The factors affecting the action of enzymes. There are various factors affecting the action of enzymes. So the first factor is temperature, second factor is pH, third factor is the concentration of the substrate. Three factors. So I will write 3 factors. So there are 3 factors. So otherwise let me write it here. There are 3 factors affecting the rate of the enzyme or action of an enzyme. Factors affecting the enzyme action. என்னென்ன factors பார்த்தா, the first factor is temperature, second factor is pH, third factor is the substrate அப்படின்னு சொல்கிறோம். அதில் இந்த temperature and pH should be optimum. Optimum அப்படின்னா என்ன அர்த்தம்? It should be moderate. Moderateாக இருந்ததுன்னா action of enzyme will be active. Action of enzyme will be active. Suppose if the temperature is tends to be optimum temperature எவ்வளோன்னு பார்த்தா it should be 40 degrees centigrade the optimum temperature. If the temperature is going to be less than 40 degrees centigrade or otherwise if the temperature is going to be more than 40 degrees centigrade, say for example 80 to 90 degrees centigrade லாம் போனா then the enzyme will get denatured. Enzymes will get denatured. Similarly, the enzyme in case of thermophilic bacteria, இல்லை thermophilic organismsல இருக்கக்கூடிய enzymes will be active at 80 to 90 degree centigrade. Okay, it can withstand hot winds and even springs. இதெல்லாம் இதோட conditions, high conditions. Hot temperature நான் மென்ஷன் பண்ணுறது தான் ஹாட் விண்ட்ஸ் அண்ட் ஸ்ப்ரிங்ஸ் அப்படின்னு நம்ம சொல்கிறோம். அது எல்லாத்தையுமே வித்ஸ்டாண்ட் பண்ணக்கூடியது தி என்சைம்ஸ் விச் ஆர் ப்ரெசென்ட் இன் தென் தெர்மோஃபிலிக் ஆர்கானிசம்ஸ். ஈவன் இன்ஆர்கானிக் கான்ஸ்டுவன்ட்ஸ் இருக்கக்கூடிய என்சைம்ஸ் மேட் அப் ஆஃப் த இன்ஆர்கானிக் கான்ஸ்டுவன்ட்ஸ் ஆல்சோ கேன் வித்ஸ்டாண்ட் அப் டு 80 to 90 degree centigrade. ஆனால் ஒரு என்சைம் நார்மலாக ஒர்க் பண்ணணும், நார்மலாக இருக்கணும் அப்படின்னா இட் நீட்ஸ் ஆப்டிமம் டெம்பரேச்சர். பிஹெச்சோட ரேஞ்ச் கூட இட் ஷுட் பி அட் ஆப்டிமம். லோ பிஹெச் அசிடிக் ரேஞ்சுக்கும் போகக்கூடாது, ஆல்கலைன் பிஹெச் ரேஞ்சுக்கும் இட் ஷுட் நாட் கோ. ஆப்டிமம் பிஹெச் ரேஞ்ச். when you go for the substrate எடுத்து வச்சு படிக்கும்போது the substrate பொறுத்த வரைக்கும் the substrate should be at a higher concentration, then the rate of the chemical enzyme action will be faster. So substrate எழுதும்போது என்ன எழுதணும்? Second point, the substrate. What you will write? Substrate should have high concentration. Substrate should have higher concentration. Whenever it has a higher concentration, then it leads to faster enzyme activity. Whenever the enzyme activity is going to be faster, then it leads to increased rate of chemical reaction. So, அப்போ substrate எடுக்கும்போது it should have a higher concentration, enzyme activity should be faster and the rate of chemical reaction will get increased. So, enzyme activity faster இருந்ததுனா the rate of the chemical reaction will be tends to be higher or otherwise it will be faster. These are factors affecting the enzyme action. And the last one is called as the enzyme inhibitors. Inhibitors are the substance which goes and binds with that of the active sites of an enzyme. Then active site நான் சொன்ன enzyme la and the active site la what has to go? The substrate. ஆனா இங்க என்ன போய் attach ஆகுது? The active site la the inhibitors, substrate inhibitors or enzyme inhibitors will goes and binds towards the active site of an enzyme. So that makes the enzymes inactive, that renders the enzymes to be inactive. You know, appropriate substrate has to get attached towards the active site of the particular enzyme, then only the biocatalytic activities will take place. Whereas here what's happening, instead of appropriate substrate, the substrate like inhibitors goes and attaches. Say for example, we take succinic dehydrogenase enzyme is there. So what is the enzyme? Enzyme is example. Now enzyme inhibitors के example எழுதுறேன். The enzyme which I am going to use is succinic dehydrogenase. இப்போ இந்த succinic dehydrogenase ன்னு சொல்லக்கூடிய enzyme க்கு use, this is an enzyme, isn't it? It is an enzyme. To this enzyme, what I have to use? I have to use the substrate. What is the substrate can be used is succinate. சொல்லக்கூடிய substrate நான் யூஸ் பண்ணேன் அப்படின்னு சொன்னால் then the Krebs cycle அப்படின்னு சொல்கிற carboxylic acid cycle the energy molecules like ATP molecules, NADPH2, everything will be getting produced appropriately. Energy will be getting liberated. Suppose succinate க்கு பதில் if instead of succinate, if malonate is going to be used, malonate. If a malonate is going to be used and this malonate will suppress the succinic dehydrogenase activity. ஏன்னா it is an inhibitor. The malonate and the succinate will have more or less similar structure. அதனால this malonate will go in disguise of the succinate and it can go and inhibit the activity of the succinic dehydrogenase. So succinic dehydrogenase is an enzyme. What is the appropriate substrate for the succinic dehydrogenase enzyme to function? Succinate is an appropriate substrate to make the enzyme to act, and this succinate is an appropriate substrate where goes and occupies the active site of the succinic dehydrogenase. But instead what happens, instead of this succinate, the malonate goes and occupies the active site of the succinate dehydrogenase. This malonate tends to be enzyme inhibitors. It tends to be what? Competitive inhibitors ன்னு சொல்லலாம், enzyme inhibitors ன்னு சொல்லலாம், substrate inhibitors சொல்றேன். It just goes and binds the active sites of the succinic dehydrogenase instead of succinate, and as a result the activity, the enzymatic activity of the succinic dehydrogenase will be getting suppressed, will be getting inhibited. So normal production of ATP and NADPH2 will not take place. அப்ப succinic dehydrogenase is an enzyme, succinate is an substrate, and malonate tends to be an inhibitors. Okay, so I have completed with this chapter the biomolecules. Enzymes பத்தி எல்லாம் சொல்லிட்டேன். Metabolism பத்தி detail I have told you. The living state na என்ன, equilibrium conditions, steady state, non-equilibrium, because this and all a chemistry oriented topic. and little difficult for you to understand, concept oriented. So you have to understand carefully and see to that you are taking down what are all the points that I have written on the board, which I have told. Everything has to be taken down in your notebook. Then only it will be helpful for you to study for the examinations. Unless otherwise in the textbook when you take, it will be in a detailed way they would have given, and it is very difficult and complicated for you to revise, go through and to understand the concepts. Okay, the simplified version which I have given, ensure that you are taking down everything what I have given in your notebook and have a glance of it, so then you can get much familiarized with the topics. Okay, so the biomolecules is over, and when time permits, again if possible, then I will deal with the physiology, otherwise we will see. Okay, so anyway, wish you all the very best, study well for your examinations, hope soon we will meet in 12th standard. Okay, thank you.